Thank you very much, Professor Kim and organizers. Welcome and thank you for coming, Edison and distinguished scientists from Jackson Laboratory. So maybe I may the only person not working on the genomics. My topic is Phenomics approach for the study of atherogenesis. So everybody here, we may agree that a key goal of biology is to understand phenotypic characteristics such as health, disease, and evolutionary fitness. And then what is phenomics? Phenomics can be defined as that the acquisition of high-dimensional phenotypic data on an organism-wide scale. Genomics and phenomics in terms of the high throughput studies in that. But genomics and phenomics are very similar, but it's quite different. In a future, we may completely understand the genomics, but phenome, we can't. I think there are several phenome projects from Arabidopsis to human. My group are working on the phenotyping of mice. Okay, there are three big projects for the phenome project. The mouse phenome database is the first phenome project in the world in Jackson Laboratory, and Europe, the EWHA, and also there are International Mouse Phenotyping Consortium, IMPC. And in Korea, we have the branch of IMPC in terms of Korea Mouse Phenotype Consortium. My group also a member of COMP. My groups are focused on the cardiovascular, not the phenome project, but phenotyping studies. I'm using the tool of the reverse genetics, the more focus on the mechanism study. So the IMPC has a very wonderful cardiovascular system phenotype for the study of phenotype. Also the Jackson Laboratory, the first founded the mouse phenome database. They also have the cardiovascular phenome project. So one scientist, is the very pagan. Everybody knows who are studying atherosclerosis, the pagan diet, is in terms of atherogenic diet. She first formulated that pagan diet for us. So she has found provide wonderful and beautiful study using the around more than 100 inbreed mouse strain with the high-fat diet. When 12 years ago, when I went to the Bahar to discuss with the Weber, she said to me, at that time, actually the Jackson Laboratory funded from the big grant from U.S. government and had built the new building for the study of ENU mutagenesis. And she partly moved the apoid knockout mice to new building. And she said to me, oh, good Taeg, is something happened. When I moving the apoid mice to new building, the lesion area was reduced. This tells us the environment is very important to show that the phenotype. We can, some scientists say that genotype and phenotype map. Here is to show the external phenotypes. In the case of cardiovascular disease, the external phenotypes could be the myocardial infarction or stroke. So to show that this external phenotype, SMP and polymorphism, is very important. So I always appreciate who are working on genomic studies. So I pick up that from information that, and then to see that the reverse genetics and making the knockout mice. And once the genes are altered, that SMP or polymorphism, whatever, the cell and tissue and whole body physiology can change it. That we can say the internal phenotypes show that internal phenotypes. These internal phenotypes can influence the diet and environment, some asbestos, and also I'm not talking about parents, the social stress, something like that, individual interactions. This external environment can influence that internal phenotypes. Those gene, and we can say the genome phenotype, the mapping to show that these external phenotypes. This is the genotype and phenotype map in the atherosclerosis. So the many genes who are working, the genomic scientists provide us, also many scientists provide us the several genes are important to show that atherogenesis. In terms of atherogenic genes, those atherogenic genes can play a pivotal role to show that atherosclerosis. And also the environment, we can say that we are usually using the high fat, high cholesterol diet to see that environmental factors. And this high fat diet can induce the obesity. And then this adipose tissue produces several cytokines, adipokines, and then can influence that atherogenesis. So once that the environmental factor and genetic factors changes, and then with tissue and cell phenotypes will be changed. The many immune cells, almost all kinds of immune cells, can infiltrate into intimal region of aorta. This is, we are saying, the plaque lesions. So when we see that inside of the plaque lesions, we can find almost all kinds of immune cells. And I will share today this T cell and monocyte, macrophage and dendritic cell can play a pivotal role to see the internal and external phenotype. So the hypothesis is quite simple. Let me say that T cell first. And CD137 is T cell costimulatory effect. Once we knock out the T cell costimulatory effect, there is a limitation to study atherosclerosis with mice. The mice inbred strain, they don't have plaque lesions in the aorta. So you have to use the background, the apolipoprotein E or LDL receptor knockout mice. It's a limitation for the study atherosclerosis. Anyway, so once knockout in this one of those knockout mice background, and then we can see that if that lesion area is reduced, these genes could be the atherogenic genes, and if once knockout these genes and those background lesion area will increase, this gene could be protective roles. And first we have to see, any here, see that in the lesion area, that gene should be in there, right? CD137, you can find that on T cell inside lesion area, and also that receptor, a ligand on the monocyte and the macrophage and dendritic cells. When you look out those in the background of APOE mice, you can find that the lesion area, total lesion area from aortic sinus to femoral artery, that lesion area was decreased, and also the cross-section lesion area was decreased. And to show that, to prove the mechanism, we are focusing on the type 1 interferon gamma, and then once look out this CD17, the producing interferon gamma will decrease, and then cellular phenotype, the recruitment into intima will be decreased, and then the lesion area will be decreased. So we have to see that the T cell have produced the interferon gamma in the lesion area. And also we confirm that, and also with the cellular internal phenotyping change. Once knock out that, the recruited the monocyte and macrophage were reduced, and the transcription level also reduced. And then percent of interferon gamma positive T cells also reduced in the background of LDL receptor and APOE, both knockout mice. And then we treated that antagonist antibody and then agonist antibody, both of them. Then we should show that the expected result there. And then to see that the bidirectional, once ligated the ligand and the receptor, the monocyte and T cells, both cells are activated. And then we can conclude that the CD137 ligation on the T cell and monocyte macrophage. Even in dendritic cells, I didn't show that dendritic cells. We know that dendritic cells either. And those bidirectional activation can augment exacerbate the atherogenesis. And the next question was, the external phenotype was, stroke or myocardial infarction. And then to see that the external phenotypes, so the plaque lesion had the final event was rupture. And this, in this stage, we are saying the vulnerable plaque. This vulnerable plaque has 4 typical cell tissue phenotypes: the large necrotic cores, thin fibrous caps, and the large number of the infiltrated macrophages in the intima, and the elaborated proteases. Once we look out that CD137 in the background of ApoE, the both 4 phenotypes we can see. And you can also summarize the other story. Once we CD137, look out that CD137 can play the role in the necrotic core, increase the necrotic core area, and also increase the protease, expression production levels, and then this CD137 can cause of the vulnerable plaque to see that to show that the external phenotypes. And the other stories in dendritic cells. So far, there are 3 dendritic cells, and classical conventional dendritic cells, and PDC, plasmacytodendritic cell, and monocyte-derived dendritic cells. We are focused on the FLT3 dependent classical dendritic cells. To see that the role of the dendritic cell in atherogenesis, we also that the liver genetics tools. So first we have to find that in the lesion area. But we can see that the both dendritic cells, all kind of dendritic cells in the lesion area. Then we try to look out the FLT3 dependent the classical dendritic cell, and once we look out remove that the classical dendritic cell, the lesion area was increased. I don't, today I don't much tell mechanisms to you, but as you can see here, the lesion area, unclassified lesion area was increased also. We can conclude that FLT3 signaling dependent dendritic cell protect against atherosclerosis. This study was done collaboratively with the Ralph Steinman. So the other stories, the environmental fact, I said to you that once we diet high fat, the adipose tissue will increase in our body. And then in monocyte macrophagy, all monocyte, our tissue, or our body of whole tissues have residential monocyte macrophage. So now scientists will agree that point. So I was focused on the monocyte macrophage in the adipose tissue. Once you diet high fats, so the monocyte macrophage should be lowers to producing adipokines. And first, this was LAM alpha. This first name was found in inflammatory journals. Some people said that the hypoxia-induced mitotic defect. Some people say LAM alpha. I would say this is the name of the gene, but I would say the LAM alpha. First, people say that adipocyte produced this LAM alpha, but I found that not the adipocyte. The macrophage in the adipose tissue produce this LAM alpha. I show that this prove that the LAM alpha producing is dependent on the high fat diet. I show that first time. When I diet high fat, we can see that increase this LAM alpha production levels. This is secreting adipokine, so we can check in the serum. And also we have we check the LDL receptor knockout mice. Once we high-fat diet, the LDL receptor knockout mice in the serum, the LAM-alpha level was increased. Also, that OB mice, the more LAM-alpha levels than the wild-type mice. Then once we again that knockout this the PIS1, and then cross them to that. Once knockout that in the background of LDL receptor knockout mouse, the lesion area was increased. This tells us that this is astroprotective proteins. And to study the mechanisms, that is really harder to the knockout mice. So we try to do transgenic mice then, though, and then this is secreting proteins. Okay, we can using that chicken beta-actin promoter. So once we knock out, making the transgenic overexpression mice, we can find that the production level of the LAM-alpha was increased as we expected. And how about the internal phenotypes? You can see the internal phenotypes. Once you cross them, the LDL receptor knockout mice, with the overexpressed transgenic, LAM-alpha was decreased the lesion area that was in LDL receptor knockout mice. So we see that to see the mechanism, we find that the cholesterol level was decreased. And what kind of cholesterol levels? The VLDL and LDL level was decreased. We prove it. And then this is transgenic mice. Many genomic study guys saying that the transgenic overexpression is sometimes positional effect, and then we have the otherwise confirm that is not the positional effect. And also the inbreed mice, the background mice, that we are making transgenic overexpression, total cholesterol, and LDL deficient background also, so that decrease the cholesterol levels. And to see that these mechanisms, we did using the microarray study, and fortunately we can find the cytochrome 7A1. This is the very important enzymes to cholesterol, to making bile acid, to remove from our body to outside. So we see that the cytochrome 7A1 expression level in transgenic mice, cytochrome 8B1 also increased. And we studied, and the kinetics studies, cytochrome 7A1 has many kinds of important transcription factors to regulate that. The cytochrome 7 is very important. And to see that what kind of transcription factors regulate the cytochrome 7A1 levels, we see that the kinetic study in vitro, and we found that LRH1. So that LRH1 should be, the SI study and the promoter study. We confirmed that this LRH1 can activate that cytochrome 7A1 when we transgenic the overexpression of LAM-alpha. So we have to see that internal and external phenotypes. That means that cholesterol should go out body. So in the background of B6 mice and LDL deficit background mice, we see that the liver bile acid level was increased, cholesterol level was decreased, and in the PICA, bile acid level was increased, and PICA cholesterol level was increased. And we can conclude that when environment factor induced that the adipose obesity, then inside the residential monocyte macrophage can produce this LAM-alpha, and this LAM-alpha can bind to hepatocyte, and then those LRH1, which is the transcription factor of cytochrome 7A1, and then this LRH1 activates the cytochrome 7A1 expression levels, and then those cytochrome 7A1s, the change to cholesterol to bile acid and go out to our body. And this is some example of the tiny example of genome study. But we can say that this is genome phenotype map in immune and vascular cell networks. Once we change it, there are some genetic alterations. For example, I said to you 3 examples. And this genotype space can influence the cell migration and the cell infiltration, and then change that internal phenotype of plaque lesion area. Also, this internal phenotype can influence the high-fat diet, and then this internal phenotype, gene space, and internal phenotypes and external phenotype, external environment can determine the external phenotypes in terms of myocardial infarction and stroke. And I always appreciate my lab members. The Jae-Woon-Chae is my first student. He's in working on Hanyang University, and also Oliver Smiths. Since I was his postdoc, we are still discussing on our study. Thank you very much again.